Mars, the red planet, has always been in the forefront of search for alien life, and while data from rovers and orbiters show that ancient Mars had liquid water and habitable conditions for microorganisms, to date, no definitive or conclusive proof of past or present life has been found on Mars. Some scientists at NASA, however, have recently claimed that NASA may have inadvertently discovered life on Mars almost 50 years ago, and then accidentally killed it before realizing what it was. After landing on Mars in 1976, the Viking landers carried biology experiments designed to detect microbial metabolism in Martian soil by scooping up samples of Martian soil and treating them with nutrients, water, and heat. Each of the Viking landers, Viking 1 and Viking 2, carried out 4 experiments on Mars. The gas chromatograph mass spectrometer, or GCMS, experiment, which looked for organic or carbon-containing compounds in Martian soil. The labeled release experiment, which tested for metabolism by adding radioactively traced nutrients to the soil. The pyrolytic release experiment, which tested for carbon fixation by potential photosynthetic organisms. And the gas exchange experiment, which tested for metabolism by monitoring how gases that are known to be key to life, such as oxygen, carbon dioxide, and nitrogen, changed surrounding isolated soil samples. The results of the Viking experiments were confusing and have continued to perplex some scientists ever since. The labeled release and pyrolytic release experiments produced some results that supported the idea of life on Mars. In both experiments, small changes in the concentrations of some gases hinted that some sort of metabolism was taking place. The GCMS also found some traces of chlorinated organic compounds, which were dismissed as terrestrial contamination from Earth. Subsequent landers and rovers have since proven that these organic compounds occur naturally on Mars. However, the gas exchange experiment, which was deemed the most important of the 4, failed to detect organic molecules, leading most scientists to eventually conclude that the Viking experiments did not detect Martian life. Almost 50 years later, the picture has changed a lot. 8 more landers and rovers have explored the Martian surface in greater detail. Thanks to the 2008 Phoenix lander, and to later confirmation by the Curiosity and Perseverance rovers, it is now known that indigenous organic compounds do, in fact, exist on Mars. They're in a chlorinated form, however, not what the Viking-era scientists expected, and it's not known whether they derive from biological processes or from some abiotic chemical reactions that have nothing to do with life. Some researchers also believe that most of the experiments may have produced skewed results because they used too much water. The labeled release, pyrolytic release, and gas exchange experiments all involved adding water to the soil samples. At the time of those landings, scientists had very little understanding of the Martian environment. Since Earth is a water planet, it seemed reasonable that adding water might coax life to show itself in the extremely dry Martian environment, but it is also possible that approach was not the right one. In very dry deserts on Earth, there are extreme microbes that can thrive by hiding in hygroscopic rocks, which are extremely salty and draw in tiny amounts of water from the air surrounding them. Such rocks are also present on Mars, which does have some level of humidity that could hypothetically sustain such microbes. If these microbes also contained hydrogen peroxide, a chemical that is compatible with some life forms on Earth, it would help them to further draw in moisture and also may have produced some of the gases detected in the labeled release experiment. The Viking landers also observed fog on Mars, which means 100% humidity. In principle, relative humidities would have been high enough in the morning and evening hours for microbes to suck up the moisture. But too much water can be deadly to these tiny organisms, as researchers have found that extreme floods in Earth's deserts in the past have killed up to 85% of indigenous microbes that could not adapt to wetter conditions. Therefore, adding water to any potential microbes in the Viking soil samples may have been equivalent to drowning them by hyperhydrating them. If the microbial life on Mars have hydrogen peroxide in their cells, it would allow them to draw water directly from the atmosphere, and the mixture would also have other advantages, such as keeping water liquid at freezing Martian temperatures, preventing the formation of ice crystals that would rupture the cells. The Viking landers touched down in the equatorial region of Mars, where the salt content of the soil is rather low. But there are a lot of hydrogen peroxide and perchlorates in the soil, and both of those compounds are very hygroscopic. If the indigenous Martian life might have adapted to their environment by incorporating hydrogen peroxide into their cells, this could explain the Viking results. The GCMS experiment heated up soil samples prior to analysis. If the Martian cells contained hydrogen peroxide, that would have killed them. Moreover, it would have caused the hydrogen peroxide to react with any organic molecules in the vicinity to form large amounts of carbon dioxide, which is exactly what the instrument detected. This composite of images from the Mastcam-Z instrument aboard the Perseverance rover shows Earth visible as a small bright dot moving from upper left to lower right, passing behind the Martian moon Phobos on July 2, 2026. This type of event is known as an occultation, from the Latin word occultare, meaning to hide. On Earth, the most common type of occultation occurs when our moon passes in front of background stars or when planets pass behind the moon. Mars has two moons, Phobos and Deimos, and Phobos is the larger and the innermost moon. Both are lumpy and irregularly shaped, and astronomers aren't sure whether they came from the asteroid belt or were formed from an ancient impact near or on the red planet. The image took careful preparation and required perseverance to be in the right place at the right time. That's because Phobos crosses the Martian sky 3 times a day, and Earth is visible for months at a stretch, but catching one directly behind the other takes planning and also a little luck. The Mars Reconnaissance Orbiter used its high-resolution imaging science experiment or HiRISE camera to capture this impact crater in Cerberus Fossae, a seismically active region of the Red Planet. Scientists matched the crater's appearance on the surface with a quake detected by NASA's InSight lander, which was about 1,000 miles away at the moment. The crater is estimated to be about 71 feet in diameter. Most of the impacts detected by InSight lander were thought to send their seismic signals through the Martian crust. But scientists concluded the energy from this impact traveled through the planet's mantle, much deeper than expected, after studying the location of the impact crater and seismic signals linked to it. Because of this finding, models of the composition and structure of the inner planet are being reassessed. This impact crater, along with others, was found with help from a machine learning algorithm developed at NASA's Jet Propulsion Laboratory in Southern California. The algorithm searched through tens of thousands of images captured by Mars Reconnaissance Orbiter's context camera in a matter of days, detecting 123 potential craters that may have occurred at the same time InSight was recording data. Traditional methods in which human scientists carefully peer over images pixel by pixel would have taken years of work to find these matches. Human scientists still had to narrow down the pool of candidate craters to 49 that matched InSight's quake data. After discovering this impact, scientists are commanding Mars Reconnaissance Orbiter to take more detailed imagery with HiRISE. This instrument is the cover for the autofocus and context imager camera aboard the Perseverance Mars rover. The Mastcam-Z instrument, also aboard the Mars rover, took these images showing movement of the cover during a test to characterize the behavior of the cover mechanism. On January 6, 2024, the cover froze in a position that prevented Sherlock, or scanning habitable environments with Raman and luminescence for organics and chemicals instrument, from collecting data. The rover team, however, found a way to address the issue so the instrument can continue to operate. Part of the Sherlock instrument, the cover is designed to protect the instrument's spectrometer and one of its cameras from dust. Sherlock is mounted on the end of the Perseverance's robotic arm, and it uses fine-scale imaging, an ultraviolet laser, and a spectrometer to search for organic molecules and minerals altered by water, helping scientists hunt for signs of ancient microbial life. Sherlock also has a calibration target with possible Mars suit materials, and it is measuring how they change over time in the Martian surface environment.